Imagine shrinking down until a single human cell fills your entire field of view. What would you see? Not an empty bubble. You would see a controlled, organized world, surrounded by a selective border, powered by molecular energy converters, directed by stored information, and constantly moving supplies from one station to another. Today, we are going inside the cell. A cell is the basic building block of living things. Some organisms are made of one cell. Others, including humans and plants, are made of trillions of cooperating cells. Cells are broadly grouped as prokaryotes, such as bacteria, and eukaryotes, such as animal and plant cells. In this tutorial, we will focus on the eukaryotic cell and treat it like a living city. First, notice the definition from the National Human Genome Research Institute. It describes the cell as the basic building block of living things and distinguishes eukaryotes by their nucleus and membrane-bound organelles. This is our map. Now, let us zoom in and follow the workflow. Step 1. The cell membrane. Think of it as the city wall, security gate, and communications network combined. It is a flexible double layer of lipids with embedded proteins. Small molecules may cross easily, while other substances need transport proteins or energy. The membrane also receives signals, helping the cell respond to its surroundings. Without this boundary, the cell could not maintain a stable internal environment. Inside the membrane is the cytoplasm. This includes the watery cytosol and the structure suspended within it. It is not just empty space. It is a crowded working environment where chemical reactions, transport, and communication happen continuously. Step 2. The nucleus. This is the cell's protected information center. It stores DNA, the long-term instruction set used to build and regulate the cell. The nuclear envelope separates that information from the rest of the cytoplasm, while pores control what enters and leaves. Inside the nucleus, the nucleolus helps produce components of ribosomes. Step 3. Protein production. Ribosomes read molecular instructions and assemble amino acids into proteins. Some ribosomes float freely. Others attach to the rough endoplasmic reticulum, or rough ER. The rough ER acts like a manufacturing flow and transport network, especially for proteins that will be exported, inserted into membranes, or delivered to other compartments.
Step 4. The Golgi Apparatus. Picture a sorting and shipping center. Proteins and lipids arrive in small membrane packages. The Golgi modifies them, labels them, and sends them to the correct destination. A cell stays organized because cargo is not delivered randomly. Step 5. Mitochondria. These organelles help convert energy from nutrients into ATP, a molecule cells can use to power transport, movement, repair, and synthesis. Their folded inner membranes create more working surface for energy-producing reactions. Mitochondria are often called the cell's powerhouses, but the important idea is not the nickname, it is the flow of usable energy. Step 6. Recycling and cleanup. Lysosomes contain enzymes that break down selected materials inside the cell. Peroxisomes help carry out oxidation reactions and can help process potentially harmful compounds. These compartments keep powerful chemistry controlled rather than letting destructive reactions spread through the cytoplasm. Step 7. The cytoskeleton. This internal framework helps the cell keep its shape, move materials, position organelles, and sometimes move the entire cell. Microtubules act like tracks. Motor proteins move cargo along them. Actin filaments help support the cell edge and generate movement. The cell is not static, it is an active logistics system. Now, compare an animal cell with a plant cell. Plant cells have a strong cell wall outside the membrane, chloroplasts that capture light energy, and usually a large central vacuole that stores water and helps maintain pressure. Animal cells do not have cell walls or chloroplasts, and their shape is often more flexible. Both cell types still rely on membranes, DNA, ribosomes, energy conversion, and coordinated transport. Here is the key lesson. An organelle is not an isolated decoration. Each one performs a specialized job, and the jobs depend on one another. DNA instructions influence proteins. Ribosomes build proteins.
The ER and Golgi process and route them. Mitochondria supply usable energy. Membranes control the traffic. The cell works because these systems cooperate. Let us test the map. Which structure stores DNA? The nucleus. Which structures assemble proteins? Ribosomes. Which organelles help produce ATP? Mitochondria. Which structure controls what enters and leaves? The cell membrane. And which system provides tracks and shape? The cytoskeleton. The next time you hear the word cell, do not picture a tiny empty dot. Picture a living city, bounded, powered, informed, supplied, recycled, and constantly adapting. That microscopic organization is the foundation of every tissue, every organ, and every living body. Understanding the cell is the first step toward understanding life itself.